in E. coli, two proteins are necessary for lactose metabolism. These include the enzyme beta-galactosidase and a carrier molecule lactose permease, which transports lactose and other galactosides into the cell. तो एक तो बीटा ग्लैक्टोसिडेज है जो कि हाइड्रोलिसिस करता है लैक्टोज की, और दूसरा जो है लैक्टोज परमिएज है विच इज रिस्पॉन्सिबल फॉर द ट्रांसपोर्ट ऑफ लैक्टोज इनटू द सेल. The existence of the two proteins was first shown by a combination of genetic experiments and biochemical analysis. First, hundreds of lac negative mutants which are unable to use lactose as a carbon source, they were isolated. By genetic manipulation, some of these mutations were moved from the E. coli chromosome to an F-lac plasmid. This is a plasmid carrying the genes for lactose utilization. तो E. coli में से उसके क्रोमोसोम में से जीन्स जो हैं वो इस प्लाज्मिड पे ट्रांसफर किए गए. Then partial diploids having the genotypes F-lac negative / lac positive or F-lac positive / lac negative, ये जो हैं ये पार्शियल डिप्लॉइड्स हैं, यानी कि इनके ऊपर दोनों लैक नेगेटिव और लैक पॉजिटिव दोनों ही अलील्स हैं. It was observed that these diploids always have a lac positive phenotype, which means they can make beta galactosidase. This shows that none of the lac negative mutants can make an inhibitor that blocks lac gene function. तो इसका मतलब है लैक नेगेटिव म्यूटेंट्स जो हैं कोई ऐसा इनहिबिटर नहीं बना रहे. Partial diploids were also constructed in which both the chromosome and the F-lac plasmids were lac negative. इस तरह के भी बनाए गए. Using different pairs of lac negative mutants, some pairs were observed to have a lac positive phenotype, while others were observed to have a lac negative phenotype. This complementation test showed that all of these mutants, initially isolated, fell into two groups, which were called lac Z and lac Y. The existence of two complementation groups was good evidence that there are at least two genes in the lac system. The lac Z gene is the structural gene for beta galactosidase. This enzyme catalyzes the hydrolysis of O-nitrophenyl beta galactoside, which is a lactose analog, and results in the production of O-nitrophenoxide, which is a yellow product. The function of the lac Y gene product as lactose permease was strongly suggested by experiments that showed that lac Z positive, lac Y negative cells cannot transport lactose into the cells, while lac Y negative, lac Z positive cells can do so. तो जिनमें lac Z positive है वो जो है वो लैक्टोज की ट्रांसपोर्ट कर सकता है. Investigators discovered a third gene which is lac A, the product of which is a beta galactoside transacetylase, which transfers an acetyl group from acetyl CoA to lactose analog. So ये जो है ये acetyl group को ट्रांसफर कर रहा है, तो ये lac A का प्रोडक्ट है. The reason that lac A was not detected at the same time as lac Z and lac Y is that its gene product, which is transacetylase enzyme, is not required for lactose catabolism. The precise role of lac A is still a matter of supposition. One hypothesis is that transacetylase detoxifies lactose analog, that would harm cells.